Inflammation is really the factor that controls the composition of immune cells that surround the tumor. There are many states of inflammation. It's not even as simple as just turning something up or down. Cancer arises from the body's own cells, and so a lot of the features of cancer are actually shared with the body's cells, and that makes it difficult. The types of immune cells and their activation states within a tumor play a critical role in whether a patient will respond or won't respond to treatments. Our primary goal is to define and target mechanisms of tumor microenvironment inflammation. The tumor microenvironment is the collection of cells that surround the tumor that determines whether the tumor grows or is restrained. We found that a lot of tumor cells are capable of participating in and even driving inflammation. Tumors evolve over months and even years prior to detection in many cases. And in this time, there's been this interplay of immune evasion and evasion of inflammatory pathways, as well as actually sometimes adaptation to those inflammatory pathways in tumors. We look for core regulators often in tumor cells, but sometimes in dendritic cells or macrophages, other innate immune cell types. We are seeking to tweak the tumor microenvironment composition, but we're really looking for the biggest levers to do so. We use a wide variety of tools, from computational to cell culture models, synthetic biology and immune engineering. Examples of these include novel kind of CRISPR-based tools to detect whether tumor cells are making immune cytokines, as well as perturbational single-cell RNA sequencing approaches. So we can take patient tumor and immune samples directly from the operating room and study them on the individual single-cell level, study their response to treatments. And the last thing we do is to seek to advance these insights to make new therapies. Some of these could include mRNA therapies, but really projects that span from early discovery of new drug targets all the way through clinical trials. I see patients every week, and so every week I go into the clinic and I'm excited about this incredible development of immunotherapies from 15 years ago to today. But we still have patients for whom the current immunotherapies don't work. Ultimately, we hope it leads to better immunotherapies and immunotherapies that work for a broader array of patients. That is really what fuels and grounds my experience and motivates the lab.